Hello, my translation fans, and hello, my good friend Scooby-Doo. So I'm glad he's here to join us. I know how much Scooby loves translation. What is translation? We use ribosomes to convert information in mRNA into the information of proteins. Wait. Remember this? DNA, RNA, protein. Yes. Uh-oh, I ran out of space. Translation requires ribosomes. Those are the guys that do the protein synthesis, the enzymatic reactions. It is made up of ribosomal RNA and proteins. It reads the mRNA information in sets of 3 bases called a codon. The template is the mRNA. tRNAs carry amino acids. So let's see how this works, people. We shall. So, let's try to make this so we can see everything. Oh, they're just trying to show us the messenger RNA, the mRNA. This is the guy that specifies what amino acids. The ribosome. This is the guy that does the work. So this is the message. The info. Need the info. And then the transfer RNA carries the amino acid. There's that little guy. This blue guy's a transfer RNA. Moving right along. I think you can read. Right? Here we go. Cytoplasm. Remember, that's where translation happens. Reads the codons. The start codon, always AUG, always calls for methionine. Stop codons. There's 3 different guys. No amino acid coded for them. It's a protein that binds in there, not a tRNA. Anywho, we set up complementary base pairing anticodons of tRNA to codons of mRNA. What? Let's walk through it. Are you with me, Scooby? Okay, let's walk through it, and you'll get right on it, I know. Okay, so translation. Here we have the mRNA, right? Here's our little sequence of mRNA right there. We have the nucleotides, the ribonucleotides, A's, G's, C's, and U's. Here is our start codon. That's where the ribosome, that's the purple guy, right? Here's our purple guy, ribosome. That's where he finds the mRNA at that first AUG. The tRNA that has a complementary anticodon base pairs to the codon. C, A and U, U and A, C and G. That's how the message in the mRNA becomes the sequence of the amino acids. This codon is on this tRNA. It's the only one that has it, and it always carries methionine. So wherever there's an AUG, a methionine is going to get stuck there. That's that little amino acid guy. What's the next sequence? GGG. Got to have a tRNA with CCC. He jumps in there. That happens to be glycine. Pops that guy together, and it continues to move all the way down. See, look, there he is. He jumped in. Goes all the way down until it reaches a stop codon. And then, hey, guess what happens at a stop codon? It stops. And that would be the last amino acid, the one before the stop codon. And that's how a message in the mRNA becomes a sequence of amino acids in the protein, right? mRNA to protein. Translation, that's what we're doing. What else? Okay, let's look a little closer. So here, right, our ribosome guy's moving that way, started at that first AUG, popped off that methionine. There's that guy up there, right? He was on here. Now, I just scribbled it. Now these 2 amino acids are linked together. This guy's going to come in. He's going to dump his amino acid. That guy leaves. Another guy comes in. And so on as this guy reads 3 by 3 by 3 all the way till that stop. So we jump ahead. Here he is. He's moved across. That last guy pops out. all those amino acids were linked together in that peptide bond, that covalent bond, and now we have that completed amino acid chain. And what do we call a completed amino acid chain? A protein. Yay! We made protein. That was our whole point. We're pretty awesome, aren't we, Scoob? Okay, I get it. So are you getting it now? Come on, Scoob. I'm doing the best I can here. Moving on and answering some questions and see if that helps you, Scoob. Okay? Are you gonna work with me here, Scoob? Okay, yes. Try to be a little bit serious, but let's go for it. Okay. So which amino acid always begins an amino acid chain? What's an amino acid chain? That's our protein. Well, how on earth would you know that? Which codon and anticodon are associated with that amino acid? Well, how would we know that? Let's take a pause and think and see what we come up with. Okay, we're back. Okay, remember we said has to start with AUG, right? That's where that ribosome grabs on, or that's where at least where it starts doing the complementary base pairing. So AUG, remember we said always specifies methionine. So every protein has to start with the methionine, at least when it's first made. Which codon, that's the one on the mRNA, and anticodon, that's the guy on the tRNA, is associated with that guy? Well, if you know the codon, you automatically know the anticodon because of complementary base pairing. A's go with U's, U's go with A's, C's go with G's. Right? So this is RNA to RNA. Never ever have any T's. Right? AGCU? Ribonucleotides? Yeah. Are you with me, Scoob? Oh boy, it's going to be rough. All right, let's move on. See if we can catch Scooby up. Each of the codons for stopping translation binds to a tRNA molecule that does not carry an amino. Oh boy. This is bad. I just found a gigantic mistake in our book. And it's not really a tRNA. It's another protein that does stopping. It's a release factor. But whatever. I guess we're non-majors. We're trying to make it simple. Doesn't have an amino acid. That is true. And so if there's no amino acid there, it can't bind onto our chain. Right? And so then that makes that guy explode. We're just going to go with that. And okay. Just going to go with it. What do you think, Scoob? Yeah, I kind of think it's a big problem, too. I'll fix it for edition 3, I promise. Okay, moving. Question 3. Given the partial mRNA sequence, what is the amino acid sequence that would be translated? So we're saying partial because this is not, we'll go ... So it's not the whole thing. So it doesn't start with an AUG because it's not the whole mRNA sequence, someplace in the middle. So we would have to use the genetic code chart to figure this out, and we would take it 3 by 3 by 3 by 3 by 3, and we'd use the chart to figure that out. But we're going to hold off on this one because we haven't done genetic code yet, so let's move right. Oh, stop it. Okay, genetic code. It's unambiguous. Each codon codes for one specific amino acid, right? So anything in the mRNA, that triplet codon like AUG always is methionine, never anything else. Not ambiguous. Genetic code is universal. That means every organism on Earth uses the same set of codons for the same amino acids. Almost. So there's some weirdo things out there that don't. But everything else, you're a frog, you are a bird, you are most bacteria even. You're an amoeba. Everybody uses the same set of codons. Same amino acids. Hello, evolution. Common ancestor. Yes. That's why we all share the same thing. We were all one single cell way back when. Woo! Redundant. 61 amino acid coding codons, right? If you do the math, 20 amino acids, triplet codon, blah, blah, blah. Only 20 amino acids. So amino acids can be coded for by more than 1 or 2 codons. So each single codon can be methionine, but some of the codons, CCC, CCU, both of these can code for the same amino acid. So it's not ambiguous, it's unambiguous. Each one of these is only that amino acid, but both can be that amino acid. Okay, let's just look at the chart. Here's our chart. The first letter of the codon, if it's a U, and the second letter is a U, and then the last letter is any of these guys in here, so UUU is phenylalanine. UUC is also phenylalanine. If you look, UUU does not exist any place else on this chart. It is unambiguous. Anytime there's a UUU in the mRNA, it makes phenylalanine. Nothing else. But UUC can also make phenylalanine. Never makes anything but that. But in this case, these two codons both make phenylalanine. Redundant. Ooh. So you can do that for all of them. G's first, U, and then U. GUU is valine. GUC is valine. GUA is valine. GUG is valine. Pretty much all of them. Any combination with GU anything is going to give you that valine. So 4 different codons code for valine. Each one of these never codes for anything else. Look for GUU any place else on this chart. It doesn't exist. And then the stop codons, right? Here, these guys, right here, there's 3 of them. That release factor that comes in and no amino acid causes chain termination. There's nothing to add to the chain, so the ribosome just explodes. Questions. How many codons code for isoleucine? Where's isoleucine? Where is that guy? Let's look. I see leucine. I don't see it. Where is he? Oh. Ah! Here he is. You guys found him before I did. How many? 1, 2, 3. These guys. AUU, AUC, AUA, all isoleucine. What about tryptophan? Can I find it before you did? Are you finding it? Right here. Trip. That's the only one. It's just one. UGG. How about leucine? Oh my gosh, leucine's up here. And all of these. 6 of them. Holy cow, people. Okay, Scoob. What do you think of that? I know, it's pretty crazy, huh? Alright. What codons are associated with asparagine? It's kind of like asparagus. 2 of them. AAU, AAC. What about serine? Here's a serine. AGU, AGC. Is that the only... Oh, serine's tricky. He's up there too. 4 of them. Whew. Holy cow. Oh. Question 3. Oh, I can't even read it. So this goes back to the other one, right? So we can, so the original, right? If we go 3 by 3 by 3, you can just figure it out. So with that ACU, AC, so A, go over this way, C, go down, U, threonine. So that's 3. CUU. So let's go C over U. U, that's leucine. Right? Here it is. It did it for us. There's another leucine. There's valine. There's proline. There's threonine. What? The question is, what if we get rid of this A? And now our first codon is CUC. And our second codon is UUC. And our third codon is UGG and UCC and C. I'm running out of room. C, UCC, CAA, and then we have that CA that falls off. So you can just go through and use a stupid little chart and figure it out. Yay! What kind of mutation is this? A deletion if we take that guy out. Yay! Okay, so here's that big overview. Scoob, how you doing? Are you good now? Alright, I'm going to have some private tutoring with my good friend Scooby. But let's finish this up. Again, DNA, double-stranded. We're just showing the one strand. We're going to show the template here. We use that in transcription. RNA polymerase reads and complementary base pairs. If there's an A, it's a U. If there's a G, it's a C. If it's a T, it's an A. See? Yes. And then we have to do it by sets of 3 to get to the amino acids. This is translation. Ribosome does the work. DNA makes RNA, makes protein. DNA, RNA, protein. It's the central dogma. It's great. We're done with this little tutorial. I hope you've learned a lot. What do you think, Scoob? All right. Best I can do. I'll keep working with him. You guys too. Thanks for listening.